Okay, good evening everyone, and thank you again for dialing in tonight. This is our, I think, third 2021 session of State of Play. You may also, I think there's a new feature on Zoom that lets you know that the meeting is being recorded, and you have to press the continue button to be able to stay in the meeting and stay connected. Right, before we start, first an acknowledgement of country. So in the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respects to the elders past, present, and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past, present and future. Right, so currently there are a number of different pathways and mechanisms that are being investigated in MND. And one of those growing rapidly and showing some great promise is the role of the immune system, and with it, its key role in neuroinflammation. So again, tonight we have two real gun up-and-coming researchers, Doctors Nicole Fewings from the Westmead Institute in Sydney, and Dr. John Lee, who's at the University of Queensland. And first up, we'll have Nicole. As previously, each presenter will talk for about 20 minutes, and then we'll have a joint Q&A at the end. So if you have questions for either presenter, just throw them at the end of the session. And you can submit your questions either through the chat or the Q&A function on Zoom, or if you're joining us through Facebook Live, then please use the chat function on there. So without hearing any more from me, we'll get this going, and please take us away, Nicole.
Thank you, Gethin. And thank you for this opportunity. It's not often you get to give a talk in your Ugg boots sitting in your spare bedroom, so thanks for that. Let's share the screen. Okay, let's go back to this. So thanks all for coming and tuning in. I have a confession to make. I'm not an expert in ALS. I generally research autoimmune conditions such as like MS and SLE. However, a few years back, our group decided to harness the skills we learned and the techniques we learned through researching those diseases and applying them to interrogating the immune system in ALS. ALS is fatal and a rapidly progressing motor neuron disease without an effective treatment. The precise mechanisms leading to ALS are yet to be determined. However, there is an increasing evidence implicating the immune system in the rate of which the disease progresses. ALS leads to motor neuron degeneration in spinal cord and brainstem and the primary motor cortex. So collectively for this talk, I'm going to be referring to these areas as the central nervous system, or CNS for short. This is a very simplified diagram of the CNS of a healthy individual. You can see that the motor neuron is happy and we have resting microglia. Microglia are resident CNS immune cells, and they are known as major players in the brain's defense and maintaining homeostasis, which means just keeping a stable environment in the CNS. At a resting state, as shown here, they have these long protrusions or arms, which basically are reaching out and sampling the environment, looking for change in the CNS. Another cell shown here that we see are astrocytes, and astrocytes provide major structural and trophic support to neurons, supplying them with nutrients, growth factors, and maintaining the blood-brain barrier, which is this layer that you can see here. So the blood-brain barrier basically keeps the immune cells out from the CNS. And what we don't see in healthy CNS is infiltration of these immune cells, thanks to the blood-brain barrier. If we move on to what has been proposed to happen at early stages of ALS, you can see that the process of motor neuron degeneration has started. These neurons send out danger signals, and the microglia sense these signals, and they begin to expand in number, and they change their morphology to become a bit more fatter and chunkier. And at this stage, these microglia are neuroprotective, and they're trying to help out. They're trying to clean up the mess, and they're secreting growth promoting and anti-inflammatory factors to help the neuron. What you also see at this stage is an infiltration of immune cells starting from the periphery into the CNS. And mostly at this stage, the immune cells are these T regulatory cells, which, as their name states, they're there to regulate the immune response. And they do this by secreting anti-inflammatory factors. And they help maintain the microglia in their neuroprotective role. So they're trying to help out. And then at some unknown stage, it all becomes too much to handle, and you get what happens in this late neurotoxic stage. So in this neurotoxic stage, the microglia expand again, and they change again, and now they have become neurotoxic and secrete pro-inflammatory factors. And these astrocytes become reactive, meaning that they can also create inflammation and actually aid in the killing of neurons. And with this, there is a reduction in the anti-inflammatory Tregs, and now there's an insurgence from the periphery of these pro-inflammatory Th1 cells and Th17 cells. And these also secrete inflammatory signals. And basically all these factors collectively increase the progression of the neurodegeneration. So when looking at a potential therapy that may harness the immune system in ALS, we need to be careful to select therapies that can give us the right balance of anti-inflammatory cells to pro-inflammatory cells. And this will require further understanding of the role of immune cells in ALS. While ideally you would like to study this directly where the action is in the CNS of ALS humans, brains and spinal cords are hard to come by. So we can do this by profiling the immune cells that are found in blood. So I'm not going to go through each individual role of the cells that are on the next few slides, but basically they're there to show you that there is evidence of immune dysregulation in the blood of ALS patients, with cells such as dendritic cells being reduced in the blood of ALS patients, and there's been some studies to show that they could potentially be dysfunctional. There's an altered ratio of the subsets of monocytes in ALS patients. Natural killer cells, which I'll talk more about later, are increased in the blood of ALS patients. B cells, there wasn't that many studies out there looking at B cells, but there was one that showed an increase in a subset of B cells called plasma cells. So when it comes to looking at the T cell compartment, there's the CD8 T cells, which are also like NK cells, have the ability to kill, and they're increased as well. And there's while the CD4s as a whole are decreased in the blood of ALS patients, there is this sway to a more pro-inflammatory Th1s, Th17s that are increased. So the goal of our lab, so within the aforementioned cell subsets, there are like subsets of those subsets, and they express externally and internally a number of proteins which can indicate what state the cell is in, and it can be indicative of the role they may play in the immune system. So the goal of our lab is to take these broader subsets, like the T cells or the B cells, and study them in depth to potentially at the end have a deep immune profile of the blood cells that are dysregulated in ALS. To do this, we use a technology known as flow cytometry, and this allows us to measure the proteins expressed by each cell. So we are able to use it to define different cell subsets. In flow cytometry, you use antibodies, they're called antibodies, and they bind to specific proteins, and they are fluorescently labeled, and each one has, like, each antibody will have a different fluorescent label, so then you can know what marker is what when you mix these. So in the end, you mix them with the blood cells, and these antibodies bind to specific proteins on or in the cell, and this mix of cells and antibodies are run on a flow cytometer. And lasers excite the fluorescently labeled cells, and it gives off a signal. So the laser excites these fluorescent, it then gives off a signal, and then is converted so that a computer can read the signal, and in a plot, it comes up in a plot like this, where each dot represents a cell. So to read these dot plots, basically you've got one antibody or marker on this axis here, our one antibody or marker on this axis, and if they're positive for this one, they'll sit here. If they're negative, they'll sit here for that one. If they're positive for this marker, they'll sit here as a cluster of dots. And if they're dually expressed both those markers, they sit here. So from this, you'll be able to define different subsets of cells. And to give you an insight in how far this technology has come in the past few years, when I started my research career 13 years ago, I was running like 8 marker panels, max maybe 10. And the panel I used in this study that I'm going to talk about today is 23 markers. So that's 23 different proteins we can read on these NK cells that we're going to look at in the end, NK cells at a time. So as I mentioned, one of the first subsets we are interested in doing a deep investigation of are natural killer cells. And they are one of our first line of defense in the immune system against infection, cancer, and cellular stress in the body. And it does this by directly killing these cells. So NK cells know what to kill by expression of different inhibiting and activating receptors on their cell surface. So with a healthy cell, it expresses one activating ligand for one activating receptor on the NK cell, and it also expresses this protein called MHC class 1, which basically tells the NK cell, hey, I belong here. So when a cell is like virally infected or a foreign sort of cell, the NK cell can kill it by this missing self killing. So with missing self killing, the target cell is missing this MHC class 1, and that tells the NK cell, I don't belong here, and the NK cell kills it. So there's another way that the NK cells can be stimulated to kill, and that's by this induced self-killing. So with this, the target cell, due to stress, etc., will upregulate these NK activating ligands. And then when that gets to a certain sort of number and it's sort of on the cell, it stimulates the NK cell to kill it. And this one is a good one to remember for later in the talk. So why look at NK cells in ALS? So originally, when we started thinking about this project, it was about a year, 2019. And the reasons that we were looking at to look at these NK cells was that they were expanded in the blood of ALS patients. That's been shown. And there was one study that showed that this increase in NK cells in ALS correlated with disease progression. And also there was an increase in this cytokine IL-15, which is a cytokine critical for NK survival and function. Then come 2020, this study was published, and this was a game changer for their involvement of NK cells in ALS. So what they found, one, they actually detected NK cells in the spinal cord and cerebral motor cortex of ALS patients, and this was not observed in their controls. And using a mouse model of ALS, they found that NK cells can directly kill motor neurons. And they do this through this NKG2D MALT1 axis. And so NKG2D is an activating receptor expressed on NK cells, and it can bind to MALT1, which is a stress-induced ligand. So this is that form of induced killing that I mentioned before. Then the NK cells in this system also release a lot of this pro-inflammatory cytokine called interferon gamma, which induces the neuron to secrete CCL2. And CCL2, where is it here? CCL2 then attracts more NK cells to the neuron. This secretion of interferon gamma also suppresses and downregulates the Treg cells that are there to help out, and it can also shape the microglia towards a pro-inflammatory or neurotoxic phenotype. In this model, when NK cells were depleted, the protective or neuroprotective microglia were there, and an increase in Tregs was seen. So we conducted flow cytometry using a 23 marker NK panel on a small cohort of ALS patients and healthy controls. I think there's about 40 ALS patients and 20 healthy controls. And like other studies, we found that NK cells were increased in ALS. We did not find a correlation with functional scores. However, in the future, we'd like to look at progression rate, and I'd like to get more time points from patients so we could see what's happening over time with these NK cell subsets. Like with any other immune cell subset mentioned in this talk, with NK cells, there are like a cytotoxic NK cell, which are probably the ones that can kill neurons, and reduced Tregs, but there's also these immunoregulatory NK cells, and they have reduced cytotoxicity, and they can have a regulatory role from the cytokines that they secrete. So it's a bit like there being pro-inflammatory T cells and Tregs, which are anti-inflammatory. There is a similar thing happening with NK cells. So when we looked at these cytotoxic or killing NK cells, we found that they were increased in the blood of ALS patients. And again, there was no correlation with functional score. And then we looked at the immunoregulatory NK cells, and we found a decrease in this subset. And again, no correlation with functional score was seen. And this finding fits with the theme that seems to be seen in ALS, where the immunoregulatory cells, which are there to dampen inflammation, are reduced in ALS. As I mentioned earlier, our plan isn't just to look at two subsets of NK cells. We want to deeply phenotype this subset. So the panel we ran has a number of different markers we want to look at. It has different activating and inhibitory receptors, different killing receptors, and just different, so we can look at different differentiation, so we can look at what stage of their sort of life cycle that the NK is at. And ultimately, we want to do what's called like high dimensional flow cytometry analysis, which will allow us to delineate many different NK cell subsets. So this uses different mathematical algorithms that are very confusing to take all the markers that we looked at and sort of put the like cells together based on the different expressions of their markers. So it's a bit like, I like to think of it like this fruit salad where it's all mixed in together, and then using this mathematical algorithms, it's called flow sum or phenograph, it will pull out cells that are like, so like this bowl of fruit salad where they're all together. So shown over here is what they call a Tisney plot. And this is just a visualization tool that is used in these sort of high dimensional flow analysis. And basically what each, I call them nodules, probably not the correct term, these little nodules on this plot represent is a different cell subset. So this is from NK cells. These are a population of total NK cells. And these little nodes are different populations of NK cells. So hopefully at the end of all this analysis, we will be able to see what NK cell subsets are dysregulated in ALS, and this could give us a better insight into the role they play in ALS. I'd just like to acknowledge my honours student from last year who ran all these subjects, and Fiona Mackay, who's no longer with the group, but came up with the idea to look at ALS, and Samantha and Jake, who helped run the experiments, and of course the patients and controls, and the funding from MND Research Australia, which made all of this possible. And I'd like to stop sharing and pass on to John up in sunny Queensland.
Thank you. Can everyone see the screen?
Yep.
Yep. All right. Thanks for that presentation, Nicole. So I'll follow on from Nicole and present something that we've been working on for about 10 years, over 10 years now, and it's just looking at the potential role of our immune system in motor neuron disease. Yep. So what is the immune system? So everyone knows that immune system is there to protect our body and fight pathogens like bacteria and viruses. So they're like a soldier of cells, like you can see here on the right. They have different roles, but their ultimate goal is to protect our body from foreign pathogens, like viruses like COVID-19. So what the immune system does is that it initiates a key response to tissue injury or infection. And these are something that we call inflammation. So these lead to the reddening and swelling that a lot of people will see when they get inflammation around their tissues. So this is our body's attempt at self-protection. So it's trying to preserve and protect our body from some kind of insult or toxic molecules. So with the immune system and our brain and spinal cord, Nicole touched a little bit on this. So in the healthy individuals, we have a brain and spinal cord, and they actually have limited exposure to our immune cells because they have this barrier or a fence that we call the blood-brain barrier or the blood-spinal cord barrier. So this barrier here actually protects our brain and spinal cord from these immune cells that can actually initiate these immune responses. But in motor neuron disease patients, what we see is that we see this leakiness in these barriers. So this barrier actually breaks and allows these immune cells to come in and actually initiate these immune responses in our brain and spinal cord. So in our brain and spinal cord, we also have our immune system, and the main cell that Nicole touched on is the microglia. So the microglia are the resident immune cells of our brain and spinal cord. So if you look at the schematic here, so they start off being very spindle-like shape. So this is when they're actually resting or very like to keep homeostasis in the brain and spinal cord. And once you get activated due to toxic stimuli or insults, you see that they lose this spindle-like phenotype and they actually go into a blob, like a circular shape. And what this does is that it actually causes the inflammations in our brain and spinal cord. So rather than the schematic here, if you look under the microscope, this is what we see. So in a healthy motor neuron disease, in a healthy spinal cord tissue, you can see that this is the microglia staining that we see. And you can see that they have these spindle-like processes, as you can see here, which is depicted by this schematic up here. But when we look at the motor neuron disease spinal cord tissues, you can see that they actually retracted a lot of these processes and become a lot more of a circular cells, which will cause inflammation. And that's what we're seeing in motor neuron disease patients. So the question that we want to ask is, what actually activates the immune system in motor neuron disease? So on the left here, we have a healthy motor neuron. And we know that due to a genetic or environmental factors, these motor neurons can get damaged or get killed. And when these motor neurons are damaged or killed, what they do is they actually release this cell debris or protein aggregates, as we know, like SOD1, TDP43, or even the C9orf72. And these cell debris and protein aggregates that's actually released by these dying motor neurons can actually lead to the activation of our immune system. So inflammation and motor neuron disease. So like I said before, so we have cell debris and aggregates released from the dying motor neurons. What this does is that it activates our immune cells in the brain and spinal cord, which is the microglia. And these microglia can be destructive by releasing toxic molecules, which will lead to these motor neurons to actually die and get damaged. So what this will does is it will actually also release cell debris and aggregates, like I said before. So what this has formed, it's formed a cycle of inflammation-mediated neuronal death. So what our immune cell in the MND is actually doing is that it's actually killing off these motor neurons. Actually dying is actually causing more motor neurons to survive by activating these microglia. So our strategy is to actually inhibit or block this activation or the destruction to actually halt the disease progression that we're seeing in motor neuron disease. And one of the targets that we're interested in, and that's actually driving these immune responses, is our complement system. So what is complement system? So complement system has been around for a very long time. So it's been discovered 100 years ago by another scientist called Jules Baudet. And complement system is like our first defense against foreign pathogen and toxic stimuli. So the complement system is a very complicated system which has cascade of molecules, like you can see here. But the one that we're interested in is this component here in the middle, which is C5, as it is the most potent molecule within the complement systems. So what this C5 will produce, it will produce a C5A, which is like a driver of a car. And then it will drive a car called C5L1. And what this does is that it will actually cause inflammation. And we know that inflammation is causing this disease progressive nature of motor neuron disease. So to test this theory out to see if C5A or C5L1 is actually playing a role in motor neuron disease progression, we wanted to use a mouse model first because you can't test it out in human patients. So a mouse model of MND. So what we use is we use some rare form due to known mutations in the genes. So in this case, we used a SOD1. So we're using a SOD1 transgenic mouse. And this actually displays a lot of phenotypes that we see in patients like muscle weakness, body weight loss, and ultimately death due to respiratory failure. So what we did with these mouse models is that we took the spinal cord of these animals and actually looked at complement system to see if it is actually activated. So on the left here, this is a healthy mouse. So this is the spinal cord. So you can see that in green is what we consider the immune cells in the spinal cord, and red is the complement activation. As you can clearly see in the healthy normal mouse, in the spinal cord, you don't see any complement activation. However, when we looked at the spinal cord of the motor neuron disease model, you can see clear difference between the healthy and the MND mouse. So you can see that there's a lot of complement that's getting activated in the spinal cord of this MND mouse. So now that we confirmed that complement is activated, what we wanted to see is we wanted to understand what kind of a role that the CAR that we talked about before, C5L1, is actually playing. So what we did is we actually utilized a genetic approach where we genetically deleted the C5L1 from these animals and looked at their lifespan and see what actually happens to the disease. So this is what we call a survival curve in our field. So you can see that in the blue, this is the lifespan of the SOD that actually has the C5L1. So you can see this line here. And then the orange is the MND mouse model that actually lacks C5L1. And you can see that it's shifting the age or the lifespan to the right, suggesting that if you genetically delete C5L1, we can actually increase the lifespan of these motor neuron disease mouse models. So that's actually great. So our strategy now is that now we've shown that genetically deleting it shows improvement in survival. We want to be actually now developing a drug that can actually target the C5L1. So we want to develop a drug to block the complement system, but because we're also working with a brain and spinal cord disease, we wanted to make sure that this drug that we're working with can actually get into the brain and spinal cord. So one of the drugs that we developed in our lab the last 10 years is this drug called PMX205. So this is a chemical structure of it. And this is the drug that we'll be using to treat these MND models. So when we treat these animals with PMX205, it's like the survival curve as like before. In the orange, we see this is the placebo. But when we give these mice the PMX205, which blocks C5L1, you can see that it's shifting this lifespan to the right again, suggesting that if you inhibit C5L1 using a drug, that we can actually increase the lifespan of these animals, suggesting that C5L1 is playing kind of a detrimental role in motor neuron disease progression. So then when we use a grip strength, as you can see here on the left, so this is to assess their muscle strength. And you can see that the placebo, which is in the orange, it declines over time. But you can see that the mouse that gets treated with PMX205, which blocks or it dampens the C5L1, you can see that it still declines a little bit, but you see there's an improvement in the motor function compared to the placebo group. So this suggests us that in addition to increasing the survival of these animals, by inhibiting or dampening the C5L1 activation in these mouse, we can actually improve muscle strength as well. So this is the video that we've got. So on the left is the placebo-treated motor neuron disease mouse model. So you can see that it's walking around quite slow. It's very slow in walking. It's wobbly as well in their gait, suggesting that it has some kind of a motor deficit. And a lot of the natural instinct of a mouse is to explore the area, which means they'll get up on their hind limbs to actually explore. But you can see in the placebo group that this animal is not able to do that. So if you look at the right, which is the PMX205 treated mouse model, You can see the movement is a lot faster than the ones on the left with the placebo. And the gait is not as wobbly as what we see in the placebo mouse that we see in the left. And you can see that it's actually getting up on the top. So this proved to us that if you dampen or inhibit the C5L1, we can actually improve the pathology like visually as well in these mouse models. So in addition to the mouse models that we've done, we wanted to also look at the driver, which is C5A, and the car, C5L1 in MND patients. And like what Nicole was saying, we can't really look at that in the brain or the spinal cord. So what we did is we looked at it in the circulation, so in the blood. So what we found on the left here is that the C5A, which is the driver of this car of C5L1, that the number of drivers for this car is actually increased in the MND patients compared to the healthy controls. And then when we also looked at the car, which is C5L1, you can see that it's also increased in the MND patients compared to the healthy control. So what this tells us that C5A and C5L1, so the driver and the car, is actually elevated in the immune cells or even circulation of MND patients, suggesting that this could actually lead to a heightened inflammation in patients, which could contribute to the actual disease progression of motor neuron disease. This is very exciting for us at the moment because we now know that there's a lot of drugs on clinical trials at the moment, or starting on clinical trials, that's actually targeting this driver C5A or even the car C5L1. So we know that AstraZeneca or Alexion is starting a phase 3, I think in Australia as well, called Ultomara. So that actually blocks the C5, which is the driver of this system. The Ra Pharma and UCB Pharma as well is using a drug called zilucoplan, which is in phase 2 and 3. That's also blocking the actual driver C5 of the system. And we have this Alcinex company, which is promoting this PMX205 that we've worked on for the last 10 years, and has actually finally gone through phase 1. So this is the drug that actually blocks the car. So it allows, so it inhibits or stops the driver getting into the car to drive these cars, I guess, to elicit an immune response. So it's a very exciting time for us looking at these immune systems in motor neuron disease. Okay, so to summarize my talk today, so I hope I showed you that our immune system do play a detrimental role in motor neuron disease. And I've shown you that the complement system, which is part of our immune system, so the CAR, C5L1, if you activate it or promote its activity, it actually contributes to the disease pathology that we see in motor neuron disease mouse models. And we also showed, that C5L1, which is the CAR, is also elevated in human MND patients in blood. And I've also shown you that PMX205, which is our selected drug that actually inhibits this CAR C5L1, can actually ameliorate MND function, MND phenotype, and improve muscle strength and extend lifespan in the MND mouse model that we used. So to finish, before we finish off, I'd like to acknowledge our lab at University of Queensland and our collaborators at School of Medical Sciences, and also our neurologists at Royal Brisbane MND Clinic, and also our funding bodies at NHMRC, FARD-MND, MNDME Foundation, and MNDRA. Thank you very much.
Great. Thank you, John and Nicole. That was a really, really sort of brilliant simplification of some horribly complex systems. I mean, the immune system is just a lot of cells, a lot of different molecules, and you guys both did a fantastic job of trying to explain how all those different cells potentially affect MND. So I have a couple of questions. First question is for both of you. Why don't we just general anti-inflammatories then work for MND? I mean, why can't you just take some ibuprofen or something a bit more powerful like an anti-TNF, which you could take for arthritis or some of the other immune-mediated diseases? Why don't they work in MND?
Are you going to go first, or?
Oh, you be my guest.
Okay. I think, like I mentioned, it's all about getting the balance of the cells that we want versus the cells we don't want. And inflammation works through all these different pathways, and your general off-the-shelf things like Panadol or Nurofen work through a certain pathway which might reduce inflammation that causes pain. But I think with ALS, we haven't even narrowed down what pathway is resulting in that inflammation. So yeah, I think by doing what we're doing and looking deeper into the immune system, hopefully we can narrow down what pathway, so then we can get better targeted therapies and better targeted sort of anti-inflammatory therapies, which is what, like, from coming from autoimmune disease background, things like drugs that they use, like modulatory therapies they use in MS, took a lot of time to develop, to find what pathway and what drug might work in multiple sclerosis. So I just think, yeah, you sort of need to know the pathway you're targeting before you can just hit it with any anti-inflammatory. Hope that answered it.
I guess if I add a little bit to that as well, is that the immune system in motor neuron disease is like a double-edged sword, I guess. So initially in the disease process, we believe that the immune system is actually playing a beneficial role rather than the detrimental role that we're actually talking about. But because of the chronic nature of the disease in motor neuron disease, and you see this chronic neuronal death that's occurring in the brain and the spinal cord, that actually is causing the immune system to become rogue, I guess, becoming what it normally does in the body, to actually switch to this thing where it's becoming a rogue and actually attacking its own neurons, like what we see in autoimmune disorders. And I guess the thing is, with a lot of the therapies, like what you're saying, Gethin, like the TNF alpha inhibitors, is that it's a bit more downstream of what the activation of the immune cells actually start off with. So if you go down with TNF alpha, you're narrowing it too much on that one specific pathway. So I believe that if you want to target immune response overall in general, you want to go a bit higher up. So this is what we're talking about, is that if you actually target these receptors, what I call the CARs, that actually elicits multiple outcomes. For example, C5AR1 or even TLR4, or all these other receptors that we know of in the innate immune system, that can actually activate the secretion and production of multiple cytokines. So I guess rather than going down and inhibiting one specific cytokine, if you inhibit a CAR or a receptor that can actually initiate responses that produces multiple cytokines, that will be more of an efficient way of inhibiting the immune response than actually going down that narrow pathway, I guess.
And does the I might have missed this, complement system interact with NK cells, or are they two fairly separate pathways? So would you need two drugs, one to inhibit complement, one to inhibit NK cells?
No, there's a lot of interplay between what we talked about as well. So the complement system is expressed on a lot of these immune cells that Nicole touched on. So if you do go in with a complement drug, it will actually inhibit multiple immune cells that we talked about. It's not as specific that one cell that I'm talking about. So microglia is a specific cell that I'm talking about in the brains, but the complement drug itself can inhibit multiple immune cells that we know of in the circulations. And C5AR1 is expressed on multiple of immune cells that we know of.
Okay, so are there other drugs, Nicole, that perhaps specifically target NK cells that you know of at the moment?
A lot of the drugs that are out there to target NK cells are actually to boost them, because there are a lot of the ones that are used for cancer. That some of the immune modulatory therapies used in MS do show, because MS has that similar balance where there's an increase in the cytotoxic NK cells and a decrease in those immunoregulatory ones. There are immune cells, there are like therapies that sort of correct that balance, but they also, a bit like John's proposed therapy, they target a number of other cells, so it's not solely NK specific. So that I know of, there isn't any, because usually they're trying to boost NK cells. There's not that any NK depleting therapies in the works that I know of.
And we have a question here. So do you have an idea what is the initial trigger that basically starts this these sort of cycle of inflammation? What is the original thing that sets off the signals that sets off the immune system? Do you have any idea?
I think it's the motor neurons actually dying. So when the motor neurons actually die, they burst open, and when they burst open, they release, like what I said, the cell debris from the actual cell from dying, and the actual the protein aggregates within those motor neurons is what's activating this immune cell. So what I was saying before as well is that in early disease process, these microglias within the brain and spinal cord is acting to mop up these cell debris and protein aggregates because it's an external factor. So it's something that's unusual in the area of the brain and spinal cord. So what the immune response does as a response is to go in and clean up that cell debris and mop up the protein aggregates. But what I'm saying is that because the motor neuron disease is such a chronic nature, if you have a chronic cycle of these motor neurons keep dying, what the microglia is doing is that it will mop it up, but it will also be targeting the neurons that's actually not dying as well. So that's actually accelerating the disease process. So the immune system, I think, is a contributor to disease progression rather than the actual onset of or the initiator of disease. So with the disease initiator, there's a lot of things that people are doing at the moment as well, right? So there's 10% familial. So you have the SOD1, TDP43, C9orf72 mutations, but you also have these 90% sporadic, and there's a lot of these research being done now that actually can lead to, these motor neuron disease, I guess, environmental factors. And I guess that initial trigger, which is the genetic or environmental, is actually causing these motor neurons to die, which is what's triggering the activation of the immune system in the brain and spinal cord.
Yeah. Okay. Yeah. I kind of missed that bit. So basically, there is a challenge that perhaps the sort of immune mediated therapies will need to be timed very carefully when you actually give them to patients. Like, obviously, with drugs like riluzole at the moment, the idea is you want to get them into the patients as quickly as you can, as early as you can in the disease to have the maximum effect. But I'm guessing that might not be the case for the immune system, that you actually want to give the immune system a bit of a chance to try and slow the disease down initially. And when it loses control, that's when you perhaps want to start adding in the immune modulators into the system. Is that a correct interpretation?
Yes, but I guess the thing is with motor neuron disease itself, the diagnosis is pretty late in patients. There is some complication with diagnosis, right? So it's a late onset diagnosis, and it's always the diagnosis is not a definite or things like that. So because the diagnosis is quite late in these patients, I guess going in with the immune modulators once you're diagnosed could be a good time to actually go in. Like it might be you're already at that later stage of process within the immune system activation, I guess. That's what I'm trying to say.
Yeah, like I showed with that early neuroprotective stage, that was only sort of seen in mouse models because it's like proposed that in humans, they're asymptomatic while that process is sort of going. They haven't showed any onset of the disease. So maybe, yeah, these immune modulatory therapies, even after there is like symptoms, it's probably the right time to start hitting them.
Okay. And another interesting question here is, if you're potentially having, you're unfortunate enough to have MND and perhaps cancer as well, and you're receiving treatment for cancer, a cancer disease, which may be obviously immune mediated, is there a potential that this could actually worsen or accelerate your MND if you're having cancer immunotherapy at the same time? Obviously, I realise you guys aren't clinicians, but
yeah, I'm like,
from a biology standpoint.
It always, because with cancer treatment, they're targeted, it depends. So like if we're going for sort of chemotherapy, which just sort of mostly targets a lot of just the proliferating cells, which therefore in turn kills cancer cells. But then if you're going for some therapies like, there's like immune modulatory ones that are, yeah, I can't really know for sure what effect it would have. yeah, I don't know, actually. If you're boosting certain parts of their immune system, like usually you're boosting their CD8 cells and their NK cells to kill tumors and that, then could you be boosting their those cells and exacerbating their MND? I don't know.
Something to be aware of.
I guess it all comes down to what the immunotherapy is, what it's actually targeting. Like, but I guess if it's an anti-inflammatory and it's stopping some of these cells that we think is playing a role in MND, yeah, then it might have a role in motor neuron disease as well. So it's complicated because your body is counteracting with cancer and motor neuron disease, I guess. So I don't know how the body will react. So,
and you addressed this a little bit with your slide about the cycle of kind of neuroinflammation. But do you see there's a kind of compounding, almost an accelerating effect as perhaps the disease progresses, as more and more motor neurons die, perhaps they're releasing more and more sort of protein fragments, and that then accelerates the... Has anyone actually specifically studied that or have got markers of it?
I'm not too sure, but we know that the immune system gets more activated at the later stages of the disease rather than like the mid stage of the disease. So it could be that there is an accelerating factor that's causing these immune cells to actually accelerate, I guess. But there are also the proliferation of these microglia within the brain and spinal cord also enhances dramatically at the later stage of the disease. So it could be that because you have a higher number of microglia, that you could be having a lot more of these effects that we're seeing. So the cycle, you might have a lot more of those cycles happening at the later stage of the disease rather than the middle or early, I guess. But I'm not too sure about what people have done or if they've actually looked into that specific markers for accelerating progression, I guess.
Okay. A sort of semi-related question is, do you know if people have looked at samples from patients that are slower progressing versus faster progressing? Obviously, you know, we see a full spectrum of disease heterogeneity. Do you know anyone who's done specific studies trying to compare the those different sort of disease types, or are there different sort of levels of perhaps immune activation between fast and slow progressors?
We intend to look at that. Like I mentioned, there was that one study that did look at, I can't even remember their name now, but they did look at the rate of progression and they found a correlation. What I tend to do with these studies is then sort of break, like looking at the cell subsets maybe in a person that is a slower progressor compared to someone who is a faster progressor. So that's in the future of my analysis, is to look at that sort of stuff. But that from the point of NK cells, I haven't seen them broken up that way.
I think there's a lot of group in America that's done a lot of this, and there's a lot of groups that look at immune cells, have done differences between slow and fast progressing. I know there's an extensive study done by Stan Eckel. He looked at T cells, so Tregs. So he said that a slower progressing patients have a higher number of Tregs compared to the fast progressing. So I guess the thing is the nature of the disease can actually change, or change the subset of immune cells that we have in our circulation, and that could be contributing to the slow or the fast nature of the disease itself. So there's a lot of research being done on that too. So.
Cool. And this is a question sort of perhaps more for Nicole. I don't know how you might think it's a bit mean, but I'll ask it anyway. So working in the sort of MS, lupus, or the autoimmune space, do you see, what other parallels do you see with MND, and do you see potential sort of crossovers? I mean, I think there's some MS drugs now are starting to be repurposed in MND, or even vice versa. How much of a crossover do you see between those diseases, do you think, especially with MS and MND?
So the classic one with sort of MS is that there's a downright, so that one of the biggest immune cells that have been looked at in ALS by even our group beforehand and all that is Tregs, and how they correlate with disease progression. And this reduced numbers of Tregs is also seen in multiple sclerosis, and I think also in SLE, because it's like breaking that the point where they can't sort of regulate, there's not enough of them to regulate the immune response. With NK cells, what we see in MS is slightly opposite to what's happening in ALS, where in the peripheral blood, we're seeing reduced NK cells and sort of a reduced NK signature. And the same thing I've seen with lupus and that. So it's sort of different in those aspects with the NK cells. With the MS, we never knew whether what we were seeing was because the NK cells are getting recruited and trafficking out, and the same with like SLEs. So sometimes what you see in the periphery isn't necessarily what's really going on at the site of where the disease is mainly happening. But yeah, there are some overlaps, and yeah, we are looking at these NK cells in SLE and MS and ALS, and the biggest difference we've seen is that there is this increase of NK cells in the periphery of ALS, and we haven't seen that for the autoimmune diseases that we're looking at.
John, have you thought about looking at your compounds in other sort of neurodegenerative diseases, or even autoimmune?
We're doing that already. So yeah, not autoimmune, but we've looked at a lot of these other neurodegenerative diseases, which do have a neuroinflammation as, you know, one of their disease processes. So we've done Parkinson's disease, we've done Huntington's disease, and we've done a lot of these brain diseases. And the common denominator that you're finding in these brain diseases is that you do see this activation of these immune cells, like the microglia, and it can be driven through this complement system that we're looking at, so the C5AL1. So it seems like it's a common process that we're seeing across all these neurodegenerative diseases. So that's what we're trying to do now.
Okay, and we got one last question before we wrap up for the evening. Do you think that perhaps if you can find early activation of the complement system, or perhaps a change in your NK cell profile might help with early diagnosis of the disease?
I think that's what everyone wants, an earlier biomarker, I guess, of disease.
Yeah.
We are trying to do that at the moment, but the thing is, like what we're saying is that with these patients, when they get diagnosed, it's already later in the disease, so we don't know what that biomarker will be earlier in the disease itself, because they're asymptomatic. So not too sure, but we are trying to do at the moment in patients is that we're trying to take longitudinal samples to actually see if there is an increase over time in patients, and then if that actually correlates with the ALSFRS scores as well. So we'll see if there is a correlation or a link between complement activation and the disease process, but I'm not too sure about the detection because I'm not yet. But I guess that's what everyone's earlier biomarker for motor neuron disease.
Definitely, yeah, it'd be really excellent if you could find something like that. Cool. Well, thank you, guys. We've come to the end of our questions now. And look, thank you both, John and Nicole, for some really excellent insights into how the immune system, what role it plays in MND. And obviously, there's some hope there for future treatments as well. And people will be watching those trial results very closely to see what happens. For everyone, the next session is going to be mid-July. And we're going to be looking more at the healthcare side of it, I think, for that talk about how we can improve current care of patients. Everyone who's attended today will receive a link to the recording, and that will also go up on our YouTube page as well for everyone to, if you want to send it around to your colleagues or watch it again. So good night for now, and please enjoy the rest of your evening, and please come back in a month or so and watch the next episode. So thank you again, John.